Hello, today we're going to talk about DNA or deoxyribonucleic acid. So DNA stands for deoxyribonucleic acid. It is so important in carrying the information for all of the activities of the cell. It's found in the nucleus, and we've kind of talked about that as we went through the parts of the cell. So this is just about the structure of the DNA. We can see that DNA is double-stranded. It is arranged in what's called a double helix, and it kind of twists upon like a ladder. The rungs of the ladder are made up of base pairing, and the sides of the ladder are repeating units of sugar, phosphate, sugar, phosphate. Here's a little history about DNA. DNA was first discovered in 1869. However, they did not feel that DNA was the genetic material. It really wasn't until 1940s that they thought that it was not. Okay, so there's a couple of experiments that we can talk about. The first one is in 1928. Frederick Griffith, he was a British guy. He was studying two main strains of bacteria, one that was harmful and one that was harmless. When he killed the pathogenic bacteria and mixed it with the harmless one that was living, some of the living harmless cells converted to the harmful one. So he came up with what they call the transforming factor. 1952, Alfred Hershey and Martha Chase showed that DNA was the genetic material. Okay, and they were working with a virus called T2 that only contained DNA and protein. So T2, the virus, infects E. coli, which is a bacteria. And T2 can program host cells to produce more phages. So what they did is they radioactively labeled DNA and the protein in the T2. They grew the virus T2 with E. coli in a solution of radioactive sulfur, and that labeled the protein. And they also grew the T2 with E. coli in a solution of radioactive phosphorus that actually labeled the DNA. So each batch of the T2 viruses infected non-radioactive bacteria. They agitated it in a blender and they shook off the loose particles of the T2 that remained outside of the cell. Then they spun the mixture in a centrifuge. Phages, because their light remained in the liquid, and that was what the virus was, and the cells were in the liquid. Then they measured the radioactive labeling that was in the pellet and the liquid. That showed that the DNA entered the cells, not the protein. So this was a very important experiment that showed that DNA was the actual genetic material. Then, in 1953, Watson and Crick determined the structure of DNA. Now, there were a lot of important people that played a part in the actual determining the structure of DNA. And they were from England, from the United States, and so forth. Okay, so a man named Donahue helped Watson and Crick figure out that the nucleotides were inside, just like Rosalind Franklin did. Okay, Pete Pauling was the son of Linus Pauling. He made friends with Watson and Crick, and Linus Pauling was actually working on the model of DNA in the United States, and the son would actually show Watson and Crick their letters that dads were writing to them. Another guy named Chargaff was figuring out the bases, and they were able to help Watson and Crick with that part of the model. Pretty much Watson and Crick didn't do any real research. They just were finding things out from other people. But they were the ones that did figure out that the structure of DNA was double-stranded, it turned, and that the base pairing was. Just to kind of show you a little bit about the bacteriophage with Hershey and Chase, this was the T2 experiment. So you could see that what a bacteriophage looks like. And this was just a little bit about the Hershey and Chase experiment. Again, they used the radioactive isotopes in order to figure out that DNA was the genetic material. And this is just showing you Watson and Crick with their model. And again, they were the ones who did get their Nobel Prize. Now, they got their information from Rosalind Franklin. And that was one of the main things. She was doing this X-ray crystal radiograph, and picture was on at her office. And this was what helped Watson and Crick figure out that the model was a spiral and the bases were in the middle with all the other information they were receiving from other people. She was not accredited with this because you can't win a Nobel Prize once you've passed away, and she had passed away prior to them getting the thing. But they kind of took all of the information from other people to figure out the structure. Again, just Rosalind and then Watson and Crick. Okay, so DNA is made up of repeating nucleotides. A nucleotide is made up of a sugar, which is deoxyribose, a base, which is a nitrogenous base, which would be A, T, G, or C, and a phosphate group. So nucleotides kind of look like this. The deoxyribose is here. It's connected to a nitrogen base and a phosphate. And this is what repeats as we see DNA models. This is just showing you what it would look like with the molecular balls and then just with the elements. So there are 4 bases. We have A, T, G, and C. A stands for adenine, T thymine, G guanine, and C cytosine. And this is what they look like. I want you to notice the size of these two. They are larger than these. Okay? So we have adenine, guanine, cytosine, thymine. Okay? So the smaller bases, the thymine and cytosine, are called pyrimidines because they only had 1 ring, where the bigger bases was called purines because they had 2 rings. And the examples of them is adenine and guanine. And a way to remember this is the saying pure as gold. So pure comes from purines. A is for adenine. Oops, I forgot the O, sorry. And G is guanine. So pure for purines, as A for adenine, oops, and G is for guanine. And just to show you the difference, the pyrimidines are smaller, which is T and C, and the purines are bigger, A and G. Again, just showing the difference. Okay, so DNA is made up of 2 long chains of nucleotides that are arranged in the spiral. So we call this a double helix. We do have base pairing occurs. We have a purine matching up with a pyrimidine. So A goes with T and vice versa, okay, and G goes with C. All right, so how I remember this is I grew up watching the A-Team with Mr. T with all those gold chains. So A-T for A-Team. And then also George Clooney. So you could figure out whatever works for you to memorize that A goes with T, T and A, and G goes with C, or C and G. This is just showing you how we have the structure of DNA. I want to point out that we have what are called these 3' and 5' ends. And I want you also to notice that they are what's called anti-parallel. Okay, the prime refers to the number of the carbons. You can see that they're all numbered. So the 3' end is here, and this is the 5' end here. And this is just showing you how DNA is made up of nucleotides. A nucleotide is a sugar, a phosphate, and a base. And DNA twists upon as a double helix or a different ladder. Okay, so as far as some cool facts, okay, a single human cell contains 6.6 billion base pairs in its nuclear DNA. It contains as much info as 600,000 printed pages of 500 words each, which is pretty much 900 large bio books full. If you laid it out, it would measure about 2 meters, and of that, there's only about 50,000 to 100,000 different genes in all those base pairs. Okay, here's another cool fact. If DNA were removed from every cell in a single human being and laid out end to end, it would stretch from the Earth to the sun and back 450 times, which is about 135 billion kilometers. So that's quite impressive as that is. Okay, so if you have any questions, please see me tomorrow. Thanks.